Hello boys, welcome back to another session of video tutorials, and this session is meant for class 11th biology students, and this is for 25th January 2021. As we all know, we are into the revision mode and we have been revising the entire syllabus with the emphasis on each chapter, and the emphasis is in the form of revision notes covering all the important concepts of the chapter, plus we have answered the test paper 1, and now third time we are doing with the test paper 2. So this test paper 2 will help us to understand second time what are the important points to be focused on the chapter cell, the unit of life. So here we go. All questions are compulsory, of course. Question 1 to 4 carry 1 marks, and 5 to 9 carry 2 marks, and question number 10 to 11 carry 3 marks. So this gives a rough idea that this particular chapter will not have mega question, major question, 5-mark question will not be there. That is the idea. So question number 1, which structure is called little nucleus? Question number 2, what is the function of contractile vacuole? Question 3, name the enzymes present in peroxisomes. Question 4, who gave the statement omnis cellular cellular? Omnis cellular cellular. Question 5 is in the form of a statement. Both lysosomes and vacuoles are endomembrane structures. Endo means inside. Yet they differ in terms of their functions. Comment on this. Next, question 6. Who proposed cell theory? Give its postulates, means main points. Question 7. Which cell organelle is known as powerhouse of the cell, and why is it so called? Question 8. What are the main functions of cell wall? Question 9. State differences between SER and RER. What is this SER and RER? Smooth endoplasmic reticulum and rough endoplasmic reticulum. Question 10. Explain the fluid mosaic model of plasma membrane. This is very much expected question from this chapter. Fluid mosaic model, and I hope you remember the diagram. Draw the diagram, label it. Question 11. Describe the structure of typical eukaryotic chloroplast. See, in this chapter, either mitochondria or chloroplast are the expected questions. And the unexpected questions, of course, is the cell membrane and the cell wall, the structure of it, and most important, unexpected one is that of cilia and flagellum. So take care of these. Now take a paper, start writing the answers, and after say 30 minutes or so, come to the next session, that is answer session, and compare your answers with the answers that I am giving. Question number 1, as expected, small nucleus is nucleolus, as it controls the functioning of nucleus. And of course you must remember, nucleolus plays a vital role during cell division, mitosis and meiosis. Remember that. Question 2, water balance or osmoregulation. This is an important concept in this chapter, osmoregulation. And there are certain organs and that play a very crucial role in osmoregulation. They are called osmoregulators. Like in our own case, the skin is an osmoregulator, kidney is an osmoregulator. So like that, in every organism there are certain important organs that control the inflow and outflow of both the solute as well as solvents. Next, question 3, catalase. ASC means enzyme, catalase. CoA, CoA reductase. Urate oxidase. Next, question number 4. Rudolf Virchow, expected question, Rudolf Virchow. And fifth, lysosomes and the vacuoles are endomembranous structures, yet these differ in terms of their functions. Lysosomes contain hydrolytic enzymes. Example, lipase, protease, which are able to digest lipids and proteins. Lipases will digest lipids and proteases will break down the proteins into amino acids, nucleic acid and carbohydrates. That's why lysosomes play an important role in breaking these materials. And vacuoles are membrane bound spaces that facilitates transport of many ions and other materials against the concentration gradient. It's a sort of osmoregulation. Okay, so vacuoles play crucial role in two things. One is the digestion, another is excretion. That's why we name them after their function as excretory vacuoles and digestive vacuoles. Next, question number 6. Sleeden and Swan. They are famous with their surnames perhaps. Sleeden and Swan gave the famous cell theory which states as follows. 1. All living things are made up of cells and cell products. Question 2 or point 2. The cell is structural and functional unit of all living organisms. Next, all metabolic reactions in the living things take place within the cell. The cell theory was later modified by Rudolf Virchow, who stated that all new cells arise from pre-existing cells. That means if there is any new cell is to be formed, that cell will be formed only from the pre-existing cell, wherein the pre-existing cell will undergo mitosis. That is the understanding. Question 7. The biological oxidation of the fats and carbohydrates release much amount of energy which is utilized by mitochondria for ATP synthesis. When required, energy is released from ATP molecules for various cell processes in cells. So mitochondria are termed as powerhouses of the cells. So they store energy, and whenever our body requires, they release ATP molecules. Since they store energy, they are called powerhouses because ATP is the energy currency. Next, question 8. Functions of cell wall. It provides a definite shape to the cell. Yes, without cell wall there is no shape. It protects inner contents of the cells very much. It protects a delicate plasma membrane present just below it. It allows the transport of various substances to and from the cell. It is doing guard duty. It prevents the cell contents from drying up. It also provides extra strength to the cell. So these are the very important functions that the cell wall will perform. Question number 9. SCR and RER. SER do not have ribosomes and it is composed of vesicles and tubules. Since they don't have ribosomes on those tubes, they are smooth. That's why the name itself is smooth endoplasmic reticulum. Whereas RER has got ribosomes on that. So the surface becomes rough. So only they are named as RER, rough endoplasmic reticulum. So RER have ribosomes on its outer surface and they are composed of cisternae, tube-like structures. SCR synthesizes steroids and lipids. Example, fat cell lipid secretory cells of the liver. Whereas RER main function is protein synthesis due to the presence of ribosomes. And SCR gives rise to spirosomes, whereas RER gives rise to Golgi bodies, vacuoles as well as lysosomes. See, very important functions it is doing. And SCR found near the cell membrane. RER found near the nuclear membrane. These are the very important differences between SCR and RER. Then comes the mega question and a very important question, expected question from this chapter. Fluid mosaic model was composed by Nicholson and Singer. Singer and Nicholson, it is famous. According to this, each phospholipids layer is bimolecular. Look at this lipid layer, two layers are there, double layered, and in between protein layer is there. And that is the reason why we are calling it as bimolecular. And hydrophilic, hydros means water, philic means love, love for the water. Hydrophilic ends are pointed towards the top and the bottom respectively. In this, proteins are of two categories. One is the peripheral proteins, extrinsic as you are seeing here, and integral or intrinsic proteins. These intrinsic proteins are here. Okay, the two layers of phospholipids that give us a membrane with hydrophilic heads oriented towards the outside and inside of the cell, where there is an aqueous environment. The fatty acid hydrophobic, phobic means fear, hydros means water, hydrophobic tails of phospholipids don't really like to get their tails wet. So they face each other, creating this hydrophobic region inside the bilayer. A function of this bilayer is to create a distinct hydrophobic region which is essential in both keeping fluidity and moderating who can get by the selective permeable membrane and enter our miniature city. In a later lesson, we will talk about who has allowed through and when. That is, it exactly performs the duty of selective permeable membrane. That is, it allows only good things that are essential for the body, and it will stop allowing the pathogens. It will try its best. And also, when the things are going out, it will allow only the dangerous things to go out and try to prevent the good things such as sugars, amino acids from going out. That's why we say selective permeable membrane. Question number 11. Chloroplast can be found in the cells of the mesophyll in plant cells, plant leaves. Okay. Chloroplasts are bounded by two membranes, double membranous, about 300 angstrom units in total thickness. Each membrane is 40 to 60 angstrom units thick. Intervening space between the two membranes is called periplastidial space, and it is around 25 to 75 angstrom units. The inner membrane is very intricately elaborated to form a system of lamellae. Internally, the chloroplast is divisible into two parts. Look at this. One is the stroma. This is the one, stroma. Colorless ground substance, stroma. And another one is grana. Now here you must remember this just like a 50 paisa or 1 rupee coin shape. That's the thylakoid. So when you keep 1 rupee coins, let's say you have got 10 1 rupee coins. Keep 1 rupee coins one above the other. 10 coins you keep. That becomes a granum. Okay. So like that, in the inner space of stroma, they just like a stack of coins, you've got stack of thylakoids. That stack of thylakoids is called granum, and many grana will be there. So something like 16 to 20 grana will be there, and all this is protected by two layers. One is the outer membrane and another is inner membrane. Also, in between this grana, there will be stroma lamellae which interconnect the grana. So this is the granum. We say this is the kitchen of the plant cell. Okay. In this place, the carbohydrate, that is starch, is stored. So grana is membrane system made of closed flattened sacs called thylakoids, filled with green pigment chlorophyll. That's why this plays very important role in photosynthesis. These thylakoids are closely packed and appears as piles of coins. I use the word stacks of coins. These structures are called grana. The arrangement can be in the form of simple parallel sacs running lengthwise, or maybe in a complex interconnecting network of the sacs. So what you need to do is draw the diagram, label it, understand the importance of each one. So this is how you need to answer. and it is always better and advisable that you substantiate your answers, particularly 3 marks and 5 mark questions.